All right, well, welcome everybody to our next in our series of webinars for Rainbow Tree Care Scientific Advancements. This is Plant Health Care and Pollinator Preservation. So start off with some housekeeping. This webinar is worth one ISA CEU, in other words, International Society of Arboriculture Continuing Education Unit. If you didn't enter your ISA certification number during the registration, you can put it into the questions box now for CEUs. If you have questions, please enter them into that questions box and we'll address them at the end of the webinar. And also just want to note, this webinar is being recorded and the recording will be available afterwards for reference. And to introduce myself, I'm Kent Honl, arborologist at Rainbow Companies, and I work out of our Minneapolis-St. Paul office in the Twin Cities in Minnesota. So my perspective is really shaped by 21 years working here in our Twin Cities area. And I'm also a ISA board certified master arborist. I've had that credential since 2007. And I serve as an adjunct faculty in arboriculture at Hennepin Technical College in Brooklyn Park, Minnesota. And my role as arborologist, going back to Rainbow Companies, is with the phenology and tracking of when pests get timed and really running the integrated pest management, plant health care side of the division of when our treatments get done and updating protocols. And that's amongst a variety of things that I'm responsible for. But I'm very glad to speak today on pollinators. The intended outcomes today that I want to touch on and bring across is give a practitioner view of pollinator issues for arborists out in the field, give an overview of the situation that we're in right now, provide a historical context of where we came from and how we got where we are, and hopefully give you some guidance in navigating challenging conversations with customers and the public out there, because this is kind of a hot button issue. And it all relates back to integrated pest management concepts that hopefully we've been applying since day one anyway. And there's this familiar concept of the disease triangle, and I would like to propose a flipped view of that as the health triangle. And if anyone out there also attended my foliar fungi management webinar a few weeks ago, I was leaning heavily on the disease triangle concept, which I always do in presentations with arborists and dealing with anything to do with plant health care and diagnostics. It's a very simple but powerful concept, so we'll spend some time on that. And then to wrap up, I want to provide some real-world examples of modified protocols that are effective in treating pests that we deal with in our market up here in Minnesota. And oftentimes, you know, other states, there's overlap with what we have in Minnesota if you're further west or south and east of us. So the principles will apply across the board to how you modify the protocols to preserve pollinators and effectively treat pest problems. So there is a growing public concern over pollinator health, and that's in the news all over the country from coast to coast. So that's why we're here talking about pollinators today. But I just want to point out this is balanced with a host of other issues that have a lot of media headlines right now too, such as climate change that affects the water distribution across the whole continent. You know, California is in a multi-year drought, and we've also got invasive species that are at the forefront of what we're dealing with in arboriculture and urban forestry. If you look at this picture, this is what's projected to change by the end of this century, the way that the water distribution, the precip happens over the course of seasons. It looks like more will come in the winters and less in the summer and fall. You know, when plants are not really able to absorb a lot of it, it might come in the form of snow or runoff when ground is frozen and not receptive. So that increases, in its turn, tree stresses. That opens the door for some of our things like, well, we've got Asian longhorn beetle, emerald ash borer, and even pine beetles out west, where climate change has lengthened the season and stressed more trees, allowed the native beetles to propagate more strongly than they ever have before. So it doesn't even have to be an introduced species. Invasive species can be native ones that break out of their usual role. But I just list all these things to provide a little balance that it's not just all about preserving pollinators, that we're trying to take care of other issues at the same time, which can kind of increase the challenge of what we're up to in the big picture. But if we look at it in a historical context, going back to some other introduced diseases, Dutch elm disease, a very important one. In the early 20th century, that was accidentally introduced on a load of logs shipped over from Europe. And in the early days, the way that we thought that was under control was by spraying with DDT. This is an actual quote that I pulled from a book on my shelf, the 1949 Yearbook of Agriculture. It says, "Feeding by bark beetles in living elm trees can be controlled by completely covering the bark surface with emulsion spray containing 2% DDT." And that emulsion spray, they were using diesel fuel in that too. So just imagine the real early stages, blunt force kind of approach to trying to control a disease. What we found out was that, only takes a few bark beetles to spread that disease if they escape from the DDT. And the off-target effects from DDT are very well known too by this point. But over time, through a lot of research and studies and the work of people in our industry, we figured out better ways to deal with Dutch elm disease rather than trying to control the vectors. We have systemic fungicides that we macro-infuse into elm trees and successfully keep them around that way. So things got better and more targeted over time. We don't have bird eggshells getting thin from DDT in the ecosystem, all that kind of thing. So this is a huge improvement. And getting more focused in my own time as a plant healthcare practitioner, starting in the early 1990s, I can remember when the protocol for controlling two-line chestnut borer in oak trees involved spraying trees 3 times with Dursban, chlorpyrifos. That's a material not even labeled for use anymore. And certainly I wasn't up in a bucket truck like in this picture. I think this is actually elm being sprayed for cankerworm. But we had spray guns that were the size of, big firearms, but on the end of a hose, shooting 60, 70 feet up into oak trees, and it was impossible to avoid off-target drift at that time, spraying that sort of material up that high. Then came the 2000s, and we got systemic insecticides like Merit, the Imidacloprid formulation, which was a huge breakthrough in reducing the targeted pesticide use. Imagine the volume of material we'd spray up into a tree 3 times a summer as compared to what we put into the base around an oak tree to control that two-line chestnut borer. And it's absorbed and distributed exactly where that insect was feeding. So now the focus really for a lot of people concerned about pollinators, they just pick up on the word systemic insecticides as being a bad thing or neonicotinoids as being a bad thing, without the context that those actually greatly reduced the overall pesticide use in the landscape. So they were an advance. And I'm not saying there's not still room for improvement, but they were a huge innovation and reduced off-target and misapplied pesticides by a wide margin. And if we didn't have the use of systemics like neonicotinoids or other things that we have options to use right now, what would some of the consequences be? If we are dealing with emerald ash borer, one of these invasive insects that's really getting a lot of attention now too, mass casualties of ash trees like we have seen in Fort Wayne, Indiana, and established that concept of the EAB death curve. The population is at some apparently minor level, and then it explodes, and it only levels off after there's 100% mortality. In other words, no more food left because they've eaten all of it. There's a number of options in slowing ash mortality, but most of the time you do need to have some targeted use of systemic pesticides to make sure that you don't end up just having a number of mass deaths without ability to keep up with removing them. Now here's just a idea, a real concrete example, a treated versus an untreated ash tree. So if you don't believe in treating some ash trees, well then who's going to be the one to climb up or go up in the bucket truck to remove the ones that die? You know, everyone who's a climbing arborist will tell you about ash trees that when they die they turn brittle almost like glass, and they become very hazardous to climb and try to remove them. So it's better at this point in EAV infested areas to either treat or remove the tree. And if there are clients that say, well, I'll just wait, I'll remove it later when it comes to my area, well, at that point you might be in that spike of the death curve, and it's better to get the tree out before all the tree services are overbooked with ash removals. So don't mean to dwell too much on EAB because we are here to talk about pollinators today, but in this whole stew of information about pollinators and insecticides and what's going on, how do we know what to believe? There's so much contradictory information that's out there floating around right now. Like last year, there was the Harvard School of Public Health that came out with a study strengthening the link between neonicotinoids and colony collapse of honeybee colonies. Then later, at the end of 2014, Huffington Post came out with an article about bee experts dismantled the Harvard study as activist science. And you know, Huffington Post, that's not exactly Fox News. You know, that's got its own slant to it. You know, that is what it is. But then more recently, University of Maryland says pesticides are not the sole culprit in honeybee colony declines. And another Entomology Today article, study finds imidacloprid safe for honeybees at realistic exposure levels. So there's a lot of back and forth about what's really happening out there. And my point in this talk is not to come down either way on does a neonic cause a honeybee collapse. I mean, it's an insecticide. It's pretty obvious. It kills bees if you put it on them. That's no great revelation there. It's, you know, my point here more is to say we're integrated pest management practitioners, and what we should be doing anyway is minimizing off-target risk. And if there is a risk, we have other tools in the toolbox. So I'll develop that idea further as we go along. But in the midst of all of this flurry of information that seems to contradict itself, we have to keep in mind this phenomenon of confirmation bias, which is the tendency for anybody that, you know, we all do this. We search for information that confirms beliefs or hypotheses we already hold. Just like I like this Far Side cartoon. The one scientist says, "Did you read my paper on confirmation bias?" And the other one says, "Yes, but it only proved what I already knew." I mean, we're all guilty of that. So, you know, we could spend all day debating back and forth about neonics and pollinators, but again, my point is just to say, with integrated pest management, we don't even really need to go there. We can just focus on the rest of the tools in our toolbox and change our protocols so we aren't putting pollinators at risk, and you know, we maintain some sort of a neutrality and a responsibility that way. And keep this in mind too, that things are always more complicated than you think they are. And I say this coming out of University of California, Santa Cruz horticulture background. So I've definitely got my hippie organic street cred. That's my background. You know, I came out of hort school very militantly organic, but after 21 years now in arboriculture, I've seen that there's another pathway. You know, we do have to use some things here and there that will kill some bugs, but that's how it is. And I even know organic growers, friends of mine, who will say they use 3 to 4 times more fossil fuel than conventional growers. In fact, there's typically on an organic farm, you'll see a propane tank on the back of a tractor like this where they're shooting flames over the field to scorch the weed seedlings out because they're not allowed to use herbicide for their organic certification. So yeah, they're not using a chemical herbicide, but they're using propane. And how is that extracted from the ground, probably with fracking and all of the secondary effects that come with that. So if you look below the surface in anything, there's always further, the devil in the details. So the problem is that our society is getting more polarized into bumper sticker level thinking, but we have to pull back and really kind of dig below the surface in anything we're doing. And as IPM practitioners and consultants, that's really what it's all about. Is I think of it as taking a consultative approach. And you start out from that basis of finding out what's important to your prospect or your customer. Just remain calm, confident, and humble, not taking a defensive approach about, well, yeah, we use some pesticides here and there, but find out what's important to them and relate to them that we take an integrated pest management approach that involves looking at the whole picture, targeting what we're using to treat, and maybe we don't even need to treat. And the thing I always emphasize too is that everything you do in life involves trade-offs, weighing the risk and benefit in a situation. And, for example, as we were looking at the elm macroinfusion slide, that requires wounding an elm tree to deliver the arbitect into the xylem of that tree tissue. So the wound, though, that's outweighed by the benefit of preventing Dutch elm disease from getting in there. Or say you're doing a pruning cut. You're making a wound, but it's better to have that dead piece removed off from there, or a limb that would otherwise peel off due to a bad attachment in the tree. So trade-offs are involved in everything. So it's always an option to do nothing too, but basically taking that big picture consultative approach, you don't come in trying to push your own agenda. You start out from the point of finding out what's important to the prospect or the client, and you're working in a partnership with them to achieve the goals on the site. And can also emphasize that in direct response to public concerns, labels have changed because of the concern over pollinators. So this is all part of our practice too that we have to take into account, and people should know that this is not just something that we're doing out of ignorance, treating these different conditions with pesticides, but that we're following labels that have changed because of the public concerns. So after finding out what's important to the client and working with them, having a real honest conversation, you can propose a solution to their plant healthcare need. And an important piece also is enrolling them as a participant in the solution. That's part of the IPM practice, because as we'll get into the disease triangle, there's a lot of ways that the client can be participating in the situation that will help influence the outcome, and they're not just depending on us to deliver a result by administering a pesticide or a fungicide. And keep in mind too, it's always an option to decline to provide service if you're just not finding that common ground. There are some people just committed to their belief or their confirmation bias that it's a bad thing to use a pesticide. Here's an example from this spring. One of our consulting arborists had a reply to a follow-up on a proposal for emerald ash borer prevention. The customer said, "I'll tell you the same thing I told Rainbow when contacted last year with a proposal for EAB inoculation." And here points towards inoculation isn't really what we're doing, but that's what the layperson's terminology is. And she said, "Thanks for contacting me. We no longer want to inoculate our ash tree, as it is our understanding that the pesticide used is harmful to non-invasive insects as well. In fact, I wish your organization did not promote the use of these chemicals. We will keep you in mind when, if the tree needs to be removed." So there's a lot going on in this brief couple of sentences. One thing that's encouraging is she says, "We'll keep you in mind if and when the tree needs to be removed." There's still some basis of a relationship there to work with them, so all is not lost. But I don't know that it's going to be very productive to try to convince them. They seem pretty committed, and it's pretty important to them, their belief that what we're using is harmful to non-invasive insects as well. So, we don't need to treat that one, and it's probably just better to walk away from that one at that point, because there's plenty of people, they're committed to one point of view. That is what it is. Just move on and don't create a headache for yourself or damage a relationship with that customer. Now as we get into disease triangle, I guess I don't mean to harp on this for everyone out there who already knows IPM, but again, it's such a powerful, simple concept. I refer to it all the time in training the consulting arborists here at the Rainbow Tree Care division up in the Twin Cities in Minnesota. And basically, to review it, a disease or a pest infestation doesn't exist until you've got three things coexisting at the same time. There has to be a host plant or a tree that's developed at the right stage in the right environmental conditions that will create the opportune moment for a pathogen to explore or to exploit the tissue on that plant. And if you just change one of these or eliminate one of these, you change the whole thing. So generally speaking, in our culture, we have sort of a got a problem, take a pill approach. So that means many times a season I get a call or an email from one of our arborists about, hey Kent, what kills this or that fungus on this tree? And they go straight to that pathogen corner of the disease triangle. And we've got whole protocol lists and treatment guides aimed at exactly that kind of information. But there's a lot of power in the other parts of the triangle too, by modifying the host or the environment too. If you don't think of that as part of the whole package, you're missing out on a lot of power as a practitioner in influencing the situation. Now, as I had said earlier on, I'm just proposing a flip of this idea of the disease triangle. What if we looked at it as the health triangle? Then what's open to our influence is increasing diversity in the landscape, using the right cultural practices so trees aren't injured or stressed to begin with. That's going to lead to minimal outbreak of pest and disease in the first place, and if we do need to use a pesticide, it'll probably be minimized to the extent we need to use it, or it will be more effective if we do use it because the other elements are taken care of. So diversity, if a client has a nice wooded back lot like this, that's great. There's multiple species of plants and trees growing there, and hopefully it's not going to be a single disease will run through and destroy a whole bunch of the life in there. If there are snags or dead trees and they don't present a hazard to buildings or structures or people in any way, it's just fine to leave some of them there as habitat and diversity for invertebrates. And one of my colleagues pointed out the other day too in reviewing this presentation, it's not just about honeybee hives. There's a lot of pollinators that are non-native insects, and they'll be the ones that set up in these deadfall pieces on the forest floor or up in standing trees. And there's lots of kinds of insects that are beneficial, not just beehives, but diversity. It's an important thing. Clients can promote that by just the way they manage their woodlot. Not everything has to be exactly scrubbed or turf areas right up to the trees. If there are trees with cavities in them, you can balance tree preservation with risk assessment. There's this basic formula: a tree with a defect plus a target is a hazard. So take that into account, that if you do a thorough risk assessment on a tree, it's near a building or play area in the backyard, just make sure that you've done your diligence on the risk assessment side. But if there's a reasonable, not high risk tree with a cavity in it, maybe you just leave that alone and there will be a beehive that sets up in there. I actually had this experience with a client of mine. She was a real nature lover and had all kinds of bird feeders and the big wooded back lot and just loved having the animals and everything around. And she was very pleased when a stray bee colony set up in a cavity in her red oak tree. And her next door neighbor was not thrilled about the bee colony being there. She thought maybe she would get stung from it, but my client just said, no, I don't want you to put any poisons out, just leave that bee colony alone. And kind of the irony at the end of all that, the red oak tree was healthy enough, it calloused over the cavity where the bees moved in, so they had to move on to another place again. And she was disappointed in that. But usually people are happy when the tree is healthy enough to callus back over. But really just again, that point is it's okay to leave some cavities in trees for some diversity and habitat for pollinators if they're not a risk, because we're talking urban forestry here too, and have to make sure we've done the good risk assessment. So if we get back to that health triangle or disease triangle, one of the points I really like to emphasize too is if you just change the host, that has a lot of power to change the disease situation too. Up here in Minneapolis-St. Paul area, everybody seems to be in love with the Colorado blue spruce, the Picea pungens, and the soil requirement for that is 4.6 to 6.5 pH, which only exists in small pockets of our metro area. And the Colorado blue spruce will grow fine until about 25, 30 years of age. And most people in the suburbs will have a automatic timed sprinkler that comes on every other day, and it hits the lower 3 to 5 feet of that blue spruce. And that's exactly where a host of fungal problems will show up, namely cytosporic canker, rhizosphere needle cast, because think about where the Colorado blue spruce comes from, right? As its name says, Colorado. There's about 10% atmospheric humidity there, and in Minnesota through the summers, 90% humidity is very common. So they just don't have an inherent resistance to the fungal diseases in our environment. They've changed the environment by moving a non-resistant tree into a humid place like this. If I could get my customers convinced to go with an eastern red cedar, that's really the only evergreen species in our region. Well, Minnesota has the reputation as the great piney northwoods, but that's really only the northern third of the state, or the northeastern third even. But down here in the farther southern stretches of Minnesota, eastern red cedar is about the only one that volunteers itself in, and that tells you that it's rugged enough to survive the conditions as they are. It takes the soil pH that we have in the metro area. And the people who did take that advice and plant eastern red cedar, they don't have to deal with any drought, insect, or fungus problems, and those trees are just growing happily, as the picture on the right shows. Really pretty much indestructible tree. So just by changing the host, huge power to not need to use fungicides to keep a Colorado blue spruce alive. Then this fits into the diversity piece again. If we had fewer little leaf lindens, we'd have fewer Japanese beetles showing up in our landscapes too. So maybe we rely more on ginkgos, Kentucky coffee trees. There's a host of other trees too that could increase the diversity. But I'm picking on a few of my least favorite species here, the Colorado spruce and little leaf lindens, but those are in very high proportions in our suburbs around Minneapolis-St. Paul. But there's a whole lot of other species out there that are interesting and exciting to use. Less reliance on pest-prone species. You can find a host of crabapples, for example, that are bred to resist scab. The prairie fire crabapple is one of them. And there's the added benefit if you're trying to be pollinator friendly. If it has flowers on it, it's a food supply for pollinators. So you win in that regard too. I'd like to keep in mind as well, it's not just pollinators that need flowering plants. There's other beneficial insects that if you have flowering plants, that diversity in your landscape, that will be providing food sources for soldier beetles and lacewings, these two insects pictured here. For part of their life, they feed on other insects that are harmful to your garden or your trees. And for other parts of their life, they're looking for pollen or nectar. So if you have something blooming April through September in your landscape, that's a rich source of food for all these other kinds of insects, not just pollinating insects. Then into the environmental corner of the health triangle, the good cultural practices. Proper watering is really important. Just a soaker hose around the base of a tree. It's a cheap investment, doesn't cost much, and you'll use a lot less water and deliver it slowly exactly where it needs to be. It won't run off. It won't get on the foliage of the tree, whether it's that blue spruce or the crabapple or what have you. It just gets in the ground where it's needed while it leaves the foliage dry, reduces the fungus disease. And the spruces that I see up here that suffer from winter burn versus the ones that come through the winter fine, the ones where in the spring I see they've got a soaker hose around them. And the homeowner was doing a soaker hose application in the fall to get them prepped for the winter. Mulch is another good cultural practice. You just win all the way around with this because it's recycling urban wood waste, which we tend to have in abundance in urban forestry. And studies have shown far higher root growth under mulched areas where trees are growing versus the turf. Turf is such a huge competitor for rooting space and water and nutrients that Gary Watson's side-by-side photos show it pretty clearly there. And it's also building humus as it decays, holds water in the soil, and prevents the mower blight. That's your #1 preventable tree disease, I believe, is the lawnmower injury. So a nice mulch area properly applied, not as a mulch volcano, but it will just keep the mowers at bay, and that alone is a huge benefit right there. Then we also look at, as a practitioner, do you really need to treat the situation? And what I've experienced quite a bit is that clients are motivated by these alarming, highly visible signs and symptoms of pest attacks, but that the dramatic pests, often they're the ones that only cause aesthetic damage, but the really deadly ones are the ones that are the stealthy killers. So put the side-by-side example, Japanese beetle, around 4th of July that typically shows up here in Minneapolis, St. Paul, and it's starting to just shred through linden trees. It's the first item on their menu. It looks really bad, but really that's not going to kill your tree. But the highly alarming symptoms, that's what gets the phone ringing, and as you go out there and you notice someone has an ash tree and you say, hey, is that a tree you want to preserve and keep around? And if so, you should treat it. And a lot of times that homeowner would say, I just am worried about these beetles. I don't really think I need to treat the ash until it gets to my neighborhood. Well, it might actually be in the tree already, laying eggs, and they'll hatch, and they work their way from the top of the crown down to the stem bit by bit, and by the time you notice the tree in decline, it's too late. And EAB is not going away once it sets up in an area. Whereas the Japanese beetle, it might spike for a few years and then almost disappear for a few years. That's how it's been going around here. But a little perspective is in order about, does it really need to be treated? If you want to keep the ash around, you need to treat that. You could forego the Japanese beetle treatment and it's not going to kill the tree. So again, it goes back to finding out what's really important to the client. Not that I would be trying to drive, achieve a certain agenda either way. It's just providing some perspective and finding out what's really important to the customer. And then we get to adjustment of treatment protocols. I had the slide in before about bee hazard directions for use and some of the label changes that have been put in place. And what the language says now is we need to apply some of these pesticides when bees are not foraging on plants. And we have to minimize exposure to bees and other insect pollinators when they're foraging on pollinator-attractive plants around the application site. Minimize drift onto beehives. In the case of the systemics that are the main focus of what I'd be talking about, you won't really have drift, but exposure is the real crux of the issue here, is what species we're applying it to, flowering trees mainly, and if there are plants around the base of them too, because where we apply this insecticide, if you have hostas or things around the base of a tree, they can absorb it, even if it's not a flower-producing tree. So just be aware of that. Just around the application site, that's the language that captures that piece. So what we want to do is avoid treating trees that are highly attractive to pollinators. First among those would be lindens. Those are just major magnets for bees of all sorts. Tulip tree, black locust, golden rain tree, and then crabapple and magnolia are two that we've got abundance of up here in Minnesota. And the soil treatment of trees that have flowering plants within the treatment zone, 1 or 2 feet from the base where the stuff is applied. Be aware of that if you've got flowering plants that someone's put around the base. And the issue too of treating ash and hemlock. We don't have a lot of hemlock here, but I know that hemlock woolly adelgid is a big threat out in the eastern states. Those trees are wind pollinated. There's not a whole lot of pollen that's going to be accessed by the pollinating insects in those cases anyway. So if it's a high value ash or a hemlock, you don't need to worry a whole lot about mass bee death by treating those with the wind pollinated species. But avoid the obvious ones like lindens and crabapples, the ones that are listed above there. So originally, just up until very recently, there was limited use for imidacloprid and dinotefuran systemic products in Oregon. That was in reaction, I'm sure, to the bee kill a couple summers ago from out there. But now, at this point, that restriction from the EPA applies throughout the USA, not just in Oregon. So imidacloprid, as in your Zytect, or dinotefuran Transtect, those are limited on any Tilia species. Little leaf lindens, basswoods, you name it. Just steer clear of those because of their strong attractiveness to pollinating insects. So when we look at our main systemics in the toolbox, there's 3 that come to mind that are in common use that I use in our plant healthcare division a lot. Lepitect, Transtect, and Zytect. So modifying the protocols, it's not too hard to do. You switch from Zytect to Transtect or Lepitect, just apply after the flowering period on the trees that do tend to be visited by these pollinating insects. And you just end up matching up, getting to know when your flowering period is done, and record that in your IPM calendar. And then you factor in how much uptake time for which pesticide you want to apply. Looking at Lepitect, it'll take 3 to 7 days for it to be fully distributed in the tree. And then following up after that, it's got a 30 to 40 day duration of effectiveness. And Transtect, 2 to 3 weeks. You have to forecast back from when your pest will attack, and after the flowering is done, hopefully you've got at least a 2 to 3 week interval before you anticipate the arrival of that pest. But once it's in there, then you've got 60 to 90 days of effectiveness. Zytect being the longest uptake time, it's corresponding with 1 year effectiveness. But that's because of that 1 year duration, that's why we restrict. We don't want to use that in the flowering trees, the ones that will be attractive to pollinators. So it's not the worst thing in the world to adjust the protocols in that way. Just get to know what species you're dealing with and when the flowering is done, and then use a Transtect or a Lepitect as needed. A huge help for me too in doing all this tracking in the past few years has been growing degree days. Big help in IPM. What growing degree days are, if you don't know, it's basically the average of the high and low temperature on a given day. Add those two together, divide by 2. The most common base temperature is 50. So you subtract 50 from the average of the high and the low for that day, and it gives you this number, the growing degree days. That's added to the degree days for all the days of the season leading up till your current date. And when I first started doing it, I tracked through the University of Minnesota Extension and a bunch of different sites, and it was just cumbersome until I just settled in on my favorite one, the Weather Channel Growing Degree Days Calculator. It's just very easy to use. You can plug in your zip code of where you are, or where you want to know the growing degree days, anywhere in the country, and put in your date brackets, and you can adjust it for the base temperature. But really, most of the time, we just go on a base 50 growing degree days. So you can use that to know where you are relative to most of these pests that when they hatch out, it's just machine-like. Go to extension websites. Here I have to give a big shout out to the extension services that help me a lot. Of course, University of Minnesota Extension right in our backyard here. We've got the Plant Disease Clinic for testing and helping out with diagnostics. And I've organized walkabouts on campus at the U of M and other local parks with a couple of the extension agents and my arborist group. We always learn a ton from them. And they're directly accessible for us with exchanging information. They learn from us, we learn from them. And we collaborate on other educational events. And then farther afield too, there's weekly emailed IPM reports I get from Ohio, the Buckeye Yard and Garden Line, and the University of Maryland Landscape IPM report. Even though those are quite a distance from us here, they still have the same, a lot of the same pests, and it gives me about a 2 or 3 week forecast ahead of when I can expect something to come up here to the frozen tundra of Minnesota. But it's been very helpful. So big kudos to them out in Maryland and Ohio, and the University of Minnesota as well. Now I move it to actual protocols. We used to use Zytex, Imidacloprid, very effectively against Japanese beetle on all the hosts that the Japanese beetle feeds. It's a surprising generalist, seems to start on lindens, go to crabapples, birch trees, and even on larch and roses, grapevines, a whole range of plants. But with the restrictions that we have now, of course, we don't want to have Zytex or Imidacloprid sitting there for a year in the plant. It'll get into the pollen potentially and affect pollinators. You know, there's, from what I've heard from U of M entomologists, there's enough to at least intoxicate bees and disrupt their navigation back to their hives that gets into pollen, but it's not enough to kill a Japanese beetle if they feed on that pollen. I mean, it gets into the leaves enough to kill the Japanese beetle. But the main point being now we need to remove that from our protocol and make an adjustment. We can still kill Japanese beetle, but we can do it in a way that it doesn't get insecticide into that pollen stream of the linden tree or other flowering trees too. And I bring it back to growing degree day timing. Through observation, I've seen over several seasons that by about 980 growing degree days, lindens are finished blooming. And through extension websites and experience, I've corroborated the Japanese beetle starts feeding at about 1090. And that's usually 4th of July week around here. So I'm ready to start treating those. So when it gets to about 1,000, I'm ready to pull the trigger on Japanese beetle treatments. That's when we get ready to spray with Tangard or permethrin right when the beetles show up. You get that quick knockdown. On the same visit, there can be treatment with Lepitect, soil applied, so that that gets uptake into the tree during that 14-day kill time that the Tangard has. And that carries you through the rest of the season up here with 30 to 40 days. Because by mid-late August, the Japanese beetles have moved on. If you're in other parts of the country where that season stretches out a bit more, maybe a second Lepitect application after about 3 weeks might be necessary to just continue that. Then at that point, end of that season, that stuff is worn off, and next year's beetles or next year's bees visiting the linden flowers will not take in any insecticide. So this is just one example. You know, it was very easy and convenient to use Zytex and it worked well, but now we just treat when they show up and use a shorter acting systemic as the follow-up, and it's still just as effective. Now Transtect hasn't really shown effectiveness against the Japanese beetle, so we've pulled that one, but we're doing pretty well with Tangard and Lepitect. I have to point out here too, roses present a special conundrum in dealing with Japanese beetle. I can remember when we treated these with Zytex, the foliage would stay perfect, but the beetles still come and feed on the petals of the flower, which is the whole point of growing roses. So what's the point of treating them with Zytect? You know, from what I've learned from U of M Extension is it gets the Zytect gets into the flowers of the roses at enough of a level to harm the bees, but not enough to kill the Japanese beetle. So with the label restrictions such as they are now, we can't really do much of anything on roses, and we're just telling customers to hand pick them into a jar of soapy water just to make sure that we're in compliance with labels. And because roses don't have one set period of blooming, like a linden tree, you know when it's done, and you can track that. Roses, for the most part, they're indeterminate, have a very stretched out bloom time. They're ornamental in the landscape. That way they're bred to provide interest for a long period of time, but that doesn't give us the luxury of a set end date of when you can apply a shorter term systemic. So in those cases, as much as the homeowner might not be pleased about it, they're kind of on their own to do other things to control the beetles. Maybe they put exclusion cloth over it or mechanical kind of barriers in that way. But it sort of ties our hands from doing systemics or even a permethrin spray on there. You're potentially harming pollinators with that 14-day residual of permethrin. Now for another concrete example, Magnolia Scale is pretty common here in our area, and the treatment required multi-stage process over more than one season. And this is another one where we were treating it with Zytect for years and had mixed results, I'd say, at best. But really the best results we've seen have been modifying the protocol to a win-win situation where it's pollinator friendly and we're getting better control of the Magnolia Scale. And the way we're going at this is horticultural oil during the dormant season, either in the spring before it comes out of dormancy or in the fall when it goes into dormancy. Then again, using the growing degree days, right at 2,000 degree days, get ready to go out and spray those crawlers with 10 guard to knock them back at that vulnerable stage. And locally we're trying Lepitect as that follow-up, kind of a similar pattern like I said with the Japanese beetle, and that's not really officially sanctioned or anything yet. It's just locally that's what we're giving a try. But if you've got that dormant oil application and then the 10 guard spray at 2,000, that's been the best delivery of results we've seen so far. Now the active period for these crawlers can stretch out to 2,800 growing degree days, which is quite a long time. During that part of the season we tend to gain about 25 to 30 growing degree days per day. So if you do the math, that's several weeks of activity. You might need to follow up with a second 10 guard or permethrin spray if you're not using Lepitect, or if you're using Lepitect, consider it experimental and just see what results you get. But that combination, it works pretty well for us in most of these cases with Magnolia Scale. So just to wrap it up now, review some of the points. Transform that disease triangle into the health triangle by modifying the host. Maybe you change the host species, increase the health of the host, reduce the environmental conditions that are promoting disease or infestation. Enroll your clients in participating in the process so that they're in charge of doing good cultural practices and mulching and modifying the irrigation. And that really takes the burden off from us as practitioners for delivering perfect results just by applying some treatments. That becomes unrealistic, and it just becomes more powerful to have the whole triangle dealt with and the client is on our side as a partner in the whole process. Then, not to be neglected too, is proper diagnosis, dosing and calibration of your equipment so you know what you're delivering. Know the tools in your toolbox, like the Lepitect, Transtect, and Zytect, how long it takes for them to distribute in the plant and the duration of their activity, and then calibrate your timing accordingly to avoid getting it into the pollen stream of the different species that are out there. Choose the systemics of the products strategically to avoid pollinator exposure. So I hope this has given you some background of how I've approached the pollinator issue. And if you have questions, we can take a look at those now. And there's still a couple of webinars coming up on downloadable apps and best management of tree injections in the next week or so here. So take part in those too, and see if we have some questions to deal with. Okay. Let me see. With stem-girdled trunks on linden. We got this from Greg Smith. Will Lepitect uptake to leaves? Really good question. When we were using Zytect on stem-girdled lindens, we were noticing in plenty of cases that it wasn't moving up that effectively, so really good call on that, Greg. I would say it's probably the same with any systemic you're trying to put into a linden. If there's stem-girdling roots, no matter what you apply, it's probably not going to get up in there. So you may have to just rely on the permethrin contact sprays at that point. That's been our experience too. And let's see, I listened to last year's pollinator webinar. They suggested beetle gone BTK. That's Bacillus thuringiensis. I am not sure about the BT for that on roses. It's possible. I guess we should note that one and maybe do some follow-up about BT and roses. So latest protocol for VLB. I'm not sure what you mean with VLB. Is something leaf beetle? But can you spray Lepitect and have it move through twig tissue into the leaves? Well, oh, viburnum leaf beetle. Yeah. Well, with any of those, just look at when the flowering is done and then apply your systemic of choice, or, you know, time it when the pest is there but the flowering is done. You know, I haven't followed viburnum. If I remember right, most of the viburnums flower fairly early. So you just have to look up the leaf beetle when they're in that active stage. And if they're feeding at the same time when it's flowering, then you're kind of stuck. And spraying Lepitect and have it move through twig tissue into the leaves. Probably, you know, I would think that would be the case. Are you talking about before leaf tissue has emerged and have it move from twig tissue? I'm not sure if that was what you're looking at, but oh yeah, I haven't used it that way before. I mean, there's a Lepitect infusible option, so you could do that. You know, do a Lepitect infusible, and you get it up into the tree that way. That's probably the best way to get distribution in there. And John Platt noted pollinators are a big deal now in Massachusetts and a hot button issue. It's the same in Minnesota. There is a lot of polarized public opinion about it, and you know, really, urban forestry, the level of stuff that we use is minuscule compared to agriculture, and the pollinator activists are taking on urban forestry because we're an easier target. I've had people actually admit this to me, that they know that we're an easier early target that they could potentially win against before they take on big ag. But really, you know, we're environmentalists too. We're saving trees for the whole ecosystem benefit that trees provide us. And we can do it responsibly, and sometimes you have to make use of some tools in the toolbox. You know, I wouldn't go to my doctor and say, antibiotics kill your gut flora and lead to super strains of bacteria, both of which are true, but would I make the leap from that to say, I want you to ban all antibiotics? You know, there's a place for them too. Like if you get some bacterial infection, most people would say, yeah, please nuke me with the ciprofloxacin. So that's the perspective I take, is, you know, there's a time and a place for it and a responsible way to use it. All right, any other questions out there? well, I guess we're covered then. So thanks everyone for attending. I hope you enjoyed this, and I enjoyed providing my background and experience, and have a great rest of the day.